Today we're going to walk through the steps of how to isolate DNA using the QIAGEN kit for isolating plant DNA. It's called the DNA Easy Plant Mini Kit. And it has simplified things over the years for us by pulling together all the different buffers that you would need into one place and making it a very stepwise process. So the first step is to get our algae, and we have our algae thawing on ice right here. That's 100 milligrams or so, and that looks pretty well thawed to me. So we'll take our micro pestle, and so we're going to go ahead and grind carefully, not to spill any out the side. So that's our physical disruption. Hopefully that has helped break open some of the cell walls. Now we can add our 400 microliters of buffer AP1. So we have our micropipetter set at 400 microliters. That a few times. This buffer is the lysis buffer that's meant to chemically digest the cell walls and cell membranes and hopefully release our DNA into solution. Now we have to add 4 microliters of RNase. RNase, what does the ACE tell you? The ACE should tell you that it's an enzyme, and the first part tells you what it should do, and that is to digest away RNA, because we're only interested in getting DNA, not RNA. So the same chemical steps that isolate DNA can also isolate RNA, so we just want to get rid of that. So we can selectively digest away the RNA without damaging our DNA with RNase. Right. And now we're going to incubate for 65 degrees for 2 to 3 minutes. Get a shot at that. At 65 degrees Celsius, that has allowed all of our membranes to melt, because if you remember, lipids melt. And now we're going to add buffer P3. Buffer P3 is intended to help precipitate proteins, but not nucleic acids. And that will help us get rid of unwanted proteins, enzymes, bad proteins that we don't want, histones that the DNA was wrapped up with, all that good stuff. So I've mixed that pretty well. We're going to set it on ice for 5 minutes. Ready? Yeah. Okay, we've been on ice for 5 minutes, and that's given the chance for our membranes, our phospholipids to re-solidify, and for our proteins to precipitate out. We're going to spin this at 14,000 times RPM for 5 minutes. You going? Okay, so we have our experimental tube with our broken down algae, in one tube, and we have a balanced tube with the same volume in it to make sure that everything is neatly balanced. We'll set our speed for 14,000 and our time for 5 minutes, and go. All right, our spin is over. We're gonna get our sample. I'm going to leave our balance there. So you can see that? There's a pellet down at the bottom and a fairly clear supernatant on top. Can you see that?
Yep.
We're going to add that supernatant to a Kaya Shredder mini column. And I'll take this out of the package, hopefully. And so this is what the columns look like. This is a disposable column. You can see a white filter matrix there. This filter is just to filter out stuff that we don't want, and our DNA will be in solution and will flow through it into our 2 milliliter collection tube. That's what's handy about having these kits, is that they provide you with these ready-made filtration columns. So I'm setting the pipetter for a little more volume than I expect to have to draw out, so I can do this in one step. Now, I definitely do not want to disturb that pellet. Let sleeping pellets lie. Leaving our pellet behind, I've got our supernatant. Just gonna put that on our Kaya Shredder column. And we will spin this for 2 minutes at max speed, which is 14,000 RPM. Okay, so we have collected our flow through. That's the material that flowed through the filter. And we are now completely done with this column, so we can toss it. Now we have to add 1.5 volumes of buffer AW1 to our flow through. And to do that, I'm going to estimate this at 500 microliters, or half a milliliter. So 1.5 times that would be 750 microliters to add. I did that in my head. So I'll set my micropipetter here. So I'm just pipetting up and down to get this well mixed with my flow through. Very gently so I don't splash it out. Alright. Now we're ready for the DNeasy mini spin column. This is a different kind of column than we used before. Looks very similar. Notice that it's a different color, so you keep them, to keep them separate. But this filter will bind to DNA. By adding AW1, we've changed the chemical composure of the buffer so that DNA will become sticky and will stick to this column, and stuff we don't want will flow through the column. So it has a limited capacity. We now have roughly, let's see, half a mil plus 750 would be 1.25 milliliters. That's more than we can fit in this chamber on this column at one time, so we're going to load 650 microliters at a time and do it several times to get all of our DNA stuck to that filter. Now we're going to spin this at 8,000 RPM for 1 minute. It should be bound to that filter, to that column. Our flow through in this case is stuff we don't want. Dump that out, and then add some more of our flow through from the first column. And we'll spin again. We have successfully spun all of our DNA onto this filter. Now we want to wash away excess ions and other particulate matters that we don't care about, leaving our DNA on it. So we're going to wash this filter twice with 500 microliters of AW2 buffer. So the AW2 buffer is designed specifically to wash away unwanted stuff, but leave our DNA attached to that filter. We're just going to spin this for 2 minutes at 8,000 RPM.
Yep.
Okay, so again, the flow through is just wash through stuff that we don't want, so we're going to dump that, add 500 more microliters of AW2, give it another good washing. And spin again. So we've finished our second wash. We're done with all of that. Our DNA is still stuck to our DNA Easy column. We're going to place this in a brand new, clean 1.5 milliliter microfuge tube. And to get our DNA back off of that filter, we're going to add 50 microliters of super duper purified water. This purified water is going to resolubilize the DNA into solution so that the last spin we do is going to wash our DNA into the flow through. And we'll let that sit for 5 minutes to make sure it has plenty of time to get all of our good DNA into solution. Take this for its final spin at 8,000 RPM for 1 minute, and that will leave us with 50 microliters of our DNA in pure water solution down at the bottom, which we can then take forward and use as the template for the PCR reaction, where we're going to actually make billions of copies of our gene of interest, the 18S ribosomal RNA gene.